Thank you for the introduction, and especially thanks for the invitation. It's an honor and pleasure for me to be here. I have no conflicts of interest to disclose. So today I'd like to discuss, my overall objective today is to discuss application of high-resolution genomic technologies for identifying genetic abnormalities in recurrent pregnancy loss, miscarriage, and couples with recurrent pregnancy loss. So miscarriage can be defined as a spontaneous demise of a pregnancy before the fetus reaches viability at about 24 weeks. Early pregnancy happens earlier and can be defined as a very early loss before 10 weeks. Miscarriage is a significant health issue because 10 to 15% of clinically recognized pregnancies end in miscarriage, and recurrent miscarriage affects about 2 to 3% of couples trying to conceive and has a significant medical and emotional impact. Both parental and fetal factors can cause recurrent miscarriage. Parental factors include balanced translocations, immune and reproductive organ dysfunction, coagulation, and other blood-related disorders. Fetal and embryo factors largely include large-scale chromosomal abnormalities. But for about 40% of couples, the cause of recurrent pregnancy loss is still unknown. So the advent of high-resolution whole genome technologies opens new possibilities for screening for and finding the genetic cause of recurrent pregnancy loss. And I'll be just providing examples for using chromosome microarrays for identifying microdeletions and microduplications, or CNV's copy number variants, in couples and their miscarriages. These are couples with recurrent loss, and also an example using next-generation sequencing for identifying single nucleotide changes, specifically whole exome sequencing. So genetic analysis of early recurrent miscarriage has advantages and disadvantages. The main advantage is that we are looking for a recurrent genetic abnormality in miscarriages typically derived from one or both parents, affecting a biologically relevant process for pregnancy development. So we are looking for a recurrent change. The disadvantage, however, is that the phenotype is not well defined, especially for early pregnancy loss. And here are the images that were given to me kindly by my colleague embryologist from Vienna, Tom Philipp. These are missed abortions. The picture was taken for a very early loss with hardly any really phenotype, and a later loss while the embryos were still in utero. So the lack of clear phenotype is a disadvantage, and the other disadvantage is that there are a large number of biological processes and genes that are relevant for early development. So when you do a whole exome screen, any gene that has a role in cell proliferation, migration, apoptosis, tissue remodeling, angiogenesis, coagulation, trophoblast endometrium interaction, and so on, can be considered a candidate. Can you, yeah. Is it better? So these are the definitive disadvantages of looking for a gene for recurrent pregnancy loss. So today I will be giving examples of genetic changes considered to contribute to recurrent pregnancy loss using chromosome arrays and exome sequencing. And these examples and the whole work is a result of my long-term collaboration with Dr. Mary Stevenson, who was the director of recurrent pregnancy loss clinic in Vancouver when we started working together, and then moved to University of Illinois in Chicago. And I also acknowledged research funding from Canadian Institutes of Health Research and Michael Smith Foundation of Health Research. So our study had a very simple concept. Couples were recruited that had multiple chromosomal and normal miscarriages, and then they had chromosome arrays, And then for those, in about 4% of cases, we found CNVs which we considered possible causes of recurrent pregnancy loss. And some of these underwent or are undergoing research, further research studies for functional analysis of the genes affected by CNVs in order to try to prove that there is truly an effect of the CNV on the development of the embryo. Unfortunately, the vast majority of these miscarriages had no diagnosis, or couples with miscarriages, there was no diagnosis, and I will describe using exome sequencing for 4 couples and their miscarriages, trying to find a cause that was not detected by microarrays, and also aiming to do further research and functional analysis for those variants. So the first example is a gene that we consider as a possible candidate for recurrent pregnancy loss that was identified using chromosomal microarrays. So this is the clinical finding in the couple that I will describe. They had 10 miscarriages. Vast majority of miscarriages were chromosomally normal, and they all had a very recurrent type of pathology, which is included placental infarcts and decidual infarcts, fibrin deposition. This was all described by the pathologists. But the bottom line, or the common denominator, was that there were vascular abnormalities in the placenta, which could have impaired the vascular supply to the placenta, and therefore the miscarriage. So for this couple and their miscarriages, we performed chromosome microarray and identified a small duplication affecting 5' end of a gene called TIM2. It was maternal. It was maternal in origin. So the red shows mom's duplication, and then 3 other panels show the same duplication found in the recurrent miscarriages. So why do we think that this could be the cause of the recurrent miscarriage? So this TIM2 gene, which stands for tissue inhibitor of metal protease 2, has a critical role in tissue remodeling of endometrium and placenta in pregnancy, specifically vascular remodeling and angiogenesis in the first trimester is relevant for pregnancy development, and it is expressed in both endometrium and placenta. It is also interesting that it is reported to be expressed only from the maternal allele in the placenta. So we can hypothesize that maybe the lack of the functional maternal copy inherited by the multiple miscarriages prevents placenta development and function and leads to miscarriage. The second, because the paternal copy would be inactive due to imprinting. The second hypothesis would be the abnormal TIM2 expression in the endometrium and or placenta results in impaired tissue remodeling and therefore recurrent miscarriage. We could test the part of the second hypothesis by doing RNA and protein expression for the miscarriages in comparison to controls, looking for the impact of the CNV on the gene that it affected. So here you can see that in this group of dots, this is the expression, RNA expression of TIM2 in controls, and then these are RNA was extracted from elective terminations, and this was to be as comparable as possible to the miscarriages. And this is the expression of the RNA in the miscarriages, showing definitely a drop. And also the protein expression showed reduction in the miscarriages in comparison to control early pregnancies, showing that TIM2, if nothing else, the CNV affected the gene in such a way that it reduced its expression in the miscarriage. Ideally, we would have really liked to have the endometrium as well, because this is the maternal CNV, to determine what effect it had on the expression in the endometrium, but had no tissue that was good enough to get RNA and protein from endometrium. So this case illustrates several points. First, CNVs of parental origin could be the cause of RPL, so recurrent pregnancy loss. So here we really are not looking for de novo changes, but we are looking for something that is recurrent and that can explain the recurrency of the miscarriage. And the second point is that the functional impact of CNV on genes can be studied in relevant tissues and parental reproductive tissues. So here it's a small advantage because, for example, when chromosome arrays are used to study children with developmental delay, and there is a very plausible candidate a gene affected by a CNV, there is never a possibility to study its effect functionally in the brain. But here we have the miscarriage tissue. We could have the miscarriage tissue, and also, if we think in advance, we can have the parental reproductive tissue, or maybe retrospectively and prospectively to test the effect of the CNV of the parental on the parental tissue if it is parental in origin. The second example I want to show is the identification of a plausible candidate gene for RPL using whole exome sequencing. As already mentioned, the whole exome sequencing focuses only on the coding part of the genome. And despite that, there is a huge number of variants that are identified, so one has to go through a process of filtering, removing the variants that wouldn't make sense or are not reasonable to include. So out of those huge number, thousands and tens of thousands of variants, which is a single copy, a single nucleotide variants and small deletions and insertions, one can remove the common variants seen in controls, non-coding variants that sometimes get sequenced, and variants not predicted to be pathogenic or conserved and do not fit the pattern of inheritance. We used for this study, we used VARSeq, which is a commercial, software that helped us identify rare, non-synonymous, deleterious with at least variants, non-synonymous meaning loss of function type of variants, deleterious with at least one of the 9 tools predicting pathogenicity, and conserved with detected by at least or confirmed by at least 3 conservation tools, and with an established role of human disease to minimize the number of those that we would not know how to interpret. So the approaches we used for these 4 couples with recurrently pregnant, the approaches we used was one of the 3 possible that are described on this slide. So the 3 approaches that one can use to look for mutations by exome sequencing relevant for recurrent pregnancy loss can be the following. So the first one is when you sequence the whole family. You sequence the couple and you sequence the recurrent miscarriages, looking for a mutation that would be for mutations that are heterozygous in the couple, so affect only one allele or one copy of the gene, but that are transmitted from, and the mutated copy is transmitted to each of the miscarriages. So the miscarriages have no normal copy of the gene, so both copies have a mutation. The mutations can be the same or called homozygous, or can be different compound heterozygous inherited from one each from each of the parents. The second approach is to just sequence one of the recurrent miscarriages and then look for the biologically relevant mutations affecting both copies of the gene in this one miscarriage, and then do a much deeper Sanger sequencing looking for the specific mutations in additional miscarriages and also the couple. And the third approach is to sequence the couple and then look for the heterozygous mutations affecting a biologically relevant gene, and then exome Sanger sequence, so use a cheaper approach to look for the mutations mutations transmitted to each of these miscarriages from each of the parents. This approach is particularly helpful if you don't have sufficient DNA from the miscarriages, and you start for exomes, which requires usually more DNA, but you start with the couple, and then only do Sanger sequencing for the miscarriages. So in the 4 couples that we studied with idiopathic or recurrently pregnancy loss, we used approach 1 and 2. So in approach 1, for approach 1, we exome sequenced 3 couples and their miscarriages. And 1 out of 3 of those families had mutations that we considered could be associated with recurrent pregnancy loss. And 1 of the 4 families had this second approach where we just sequenced 1 miscarriage and then Sanger sequenced the rest of the family to confirm the inheritance pattern that we would expect. And this 1 family, 1 candidate gene with mutations inherited from mom and dad was identified as a candidate. And I will mention a few words about those genes in a second. So overall, although this is a very small pilot study of 4 recurrent pregnancy loss couples, 50% of them had mutations in candidate genes that could be associated with recurrent miscarriage. So this is the result of approach 1 in a family where everybody was sequenced, like here. And the gene that was identified as a candidate is called dynein cytoplasmic 2 heavy chain 1 gene, and both. So here we can see that dad had a mutation, a green mutation, and mom had a different mutation in the same gene, a blue mutation, and miscarriage 1 had the green and the blue mutation, and miscarriage 2 had the same pattern. So 2 different mutations affecting the 2 copies of the gene were found to reoccur in this family. So why would this gene and not others be relevant? Because it is involved in a process that is important for cilia generation and maintenance, and cilia are critical for early development, and their defects are associated with association with many abnormalities of skeletal, renal, liver, retinal, and so on, and reported both to be viable and lethal. And in the mouse, the knockout of this gene causes mid-gestation lethality, and also the very abnormal cilia morphology. So this is from the paper where they used the knockout of the gene with cilia not developing normally and the mouse embryo not developing normally. So a plausible candidate for recurrent pregnancy loss in this family. Using the second approach, we identified a different gene in the 4th family. So this is the approach. So just 1 miscarriage was sequenced, and then using all the filtering and the bioinformatics tools, 2 mutations were identified in a gene called LX15, and then Sanger sequencing was for those specific mutations was performed for another miscarriage, and also for the couple, confirming the same pattern in the second miscarriage, and then the parents each had one mutation. So again, why this gene? Because it's an oxidizing enzyme expressed in placenta, and also regulates immunity and inflammation. In humans, this regulation of this gene is associated with inflammation and placental dysfunction in preeclampsia, abnormal oxidative stress response, and angiogenesis. And overexpression in the mouse showed reduced fertility with increased abortion rate, suggesting that it can be linked with the recurrent pregnancy loss. So one can look for individual genes as causes of recurrent pregnancy loss, but one can also use bioinformatic tools to determine how does the pool of genes with conserved, deleterious, rare variants affect how could it affect the biological pathways and just the diseases. So this is the study again from those 4 couples who had exome sequencing and their miscarriages, looking for this general pattern of abnormal pathway and biological processes. So what one does in this type of analysis is to identify genes with conserved, deleterious, and rare variants, which was 54 for the 7 miscarriages with studied by whole exome sequencing, and then bioinformatically determine which pathways, biological pathways, and diseases are associated with these 54 genes, more than it would be for any number of other randomly selected genes. So these are the pathways that were enriched for the 54 genes with the variants identified by whole exome sequencing of 7 miscarriages. So in red are the ones that are also seen in the genes that have conserved, deleterious, and rare variants from the couples. So this is then the first line would be genes from the miscarriages, genes from the couples, and the enrichment is the same. And the disease association, again, the enriched disease is ciliary motility disorder. It's not surprising that they are the same, almost the same, because we did not this was not based on the inheritance, but any variant that was deleterious, conserved, and rare was considered in this bioinformatic analysis. What is probably more relevant is to compare the finding with enriched pathways from the 141 positive control genes, which would be those that were identified in PubMed and the Human Genome Mutation Database that are clearly associated with pregnancy loss and abortion. So the same pathway is the complement and coagulation cascade pathway, and then the disease is not unexpectedly are abortion, habitual, and spontaneous, and pregnancy complications, because these 140 genes were selected specifically to be associated with pregnancy loss. Now this has died. And then finally, we compared the enriched pathways and diseases with those that you would get if you do whole exome sequencing of normal subjects. So there was no enrichment, and then the diseases were more or less random, and then we selected randomly 50 genes, and there was no pathway enrichment. So the complement and coagulation cascade seems to be, so the genes from this pathway seem to be affected by variants more than by chance in our miscarriages and couples, and the complement cascade has importance for pregnancy because it is important for host defense against invading organisms, and initiates inflammation and tissue injury, and the bottom line is the suboptimal immune response, which could easily lead to pregnancy loss. And the other cascade is a coagulation cascade, which the dysregulation of which could lead to blood clots and pregnancy loss. And as I mentioned, the ciliary function and the ciliary motility disorders can be of relevance for pregnancy loss because of the importance of cilia in early development. So the studies that are currently taken in order to find the cause of recurrent miscarriages using next generation sequencing are very rare, really. One can kind of group them by looking at the studies that enrolled couples and miscarriages, or did exome sequencing of couples and miscarriages, and those that did exome sequencing of the couple or the women with recurrent pregnancy loss. So on the left slide, on the left side of this slide, you see that there are probably less than 10 publications where specifically one would sequence a small number or a single family with recurrent pregnancy loss. And typically these would be somewhat later losses with some phenotype. And then there were also a few studies where a larger number of consanguineous families had exome sequencing that had recurrent fetal loss, and it was reported that 50% of those would had a pathogenic or likely pathogenic variant. On the other side of the slide are studies that involved exome sequencing of a couple or women. So on the top is a study just briefly described of exome sequencing of women with recurrent pregnancy loss. And then although exome sequencing was done in this study, only variants were searched in 234 pregnancy-related genes, and then in 50% of those 49 women, there was a variant that the authors considered as a potentially important for the recurrent pregnancy loss. And then there were two studies, somewhat different, where the couples were sequenced. In one study, 50 couples had exome sequencing, and the couples had 2 or more miscarried pregnancy losses, but they had fetal anomalies described, and then 62% of those, the authors felt that they made a genetic diagnosis for the losses. And the most recent approach just presented at the American Society of Human Genetics meeting a few weeks ago in San Diego was to use the whole genome sequencing, so-called low-pass or low number of reads type of whole genome sequencing on close to 1,000 couples with 2 or more pregnancy losses. And it revealed that 2.5% of those couples have balanced rearrangements or CNVs not identified by karyotyping, which means that by this approach, typically it's quoted that 3% of couples with recurrent loss have a visible chromosomal balanced rearrangement, but now we are adding about the same number of couples if we are using if one can use this whole genome sequencing approach as the authors did. So there's lots of potential for exome sequencing in recurrent pregnancy loss, but inevitably there are challenges, as many times for when one uses exome sequencing, and here especially if there is very little phenotype, is the variant really causative? And our judgment now is made mainly on bioinformatics, the animal models, or how the gene functions postnatally, its expression, disease association. However, we know very little of the prenatal effect, and this variant may be lethal, and we have never observed it before postnatally. And also an even more difficult issue is when to take action when we start doing exome sequencing for early miscarriages and identify something that is potentially causative, and when do we suggest that there is screening for embryos for this potentially pathogenic variant causing recurrent pregnancy loss. Nevertheless, hopefully the number of studies will increase and inform us better of how to proceed, and certainly teach us so the directions that I think are important is to increase the number of clinically well-defined families with early recurrent pregnancy loss and study the bi-whole exome sequencing. And then to catalog those variants, parental and fetal, in a database, because we have no databases that would be really directed to the prenatal world. And then, very importantly, collect and catalog obstetric history necessary pathology findings and phenotypes, which sometimes, especially from the lab's perspective, is not always, well, it's not always cataloged, but it's critical. And also the future directions, if whenever we have an opportunity to study the functional effect of the candidate variant in pregnancy tissue, and as I said, we can also sometimes be able to use the parental reproductive tissue in our investigations. And I'd like to finish here by thanking the key collaborators, the obstetrician and Mary Stevenson, and a colleague embryologist from Vienna, Tom Philip, my research lab, and the funding agencies. Thank you for your time and attention.